A very good morning to everyone and welcome to ARVO. My name is Tian Wong. I'm the Clinical Epidemiological Research Trustee, and I'm very happy for all of you to join us today for a very special occasion. I'd like to review the history of the Proctor Medal Award. This award was established in 1949 as a memorial to Dr. Francis I. Proctor, an ophthalmologist who conducted extensive research on the etiology and treatment of trachoma. This award honors outstanding research in the basic or clinical sciences as applied to ophthalmology. The 2024 Proctor Medal Award, as I'm sure you are aware, and I'm very pleased to announce, is presented to Dr. Emily Chiu, the Director of the Division of the Epidemiology and Clinical Applications at the National Eye Institute, National Institutes of Health. Congratulations, Emily. Later on, we will review some of her accomplishments, but she has essentially designed, conducted, and completed large clinical trials and epidemiological research in many different diseases, including, of course, AMD, of which she'll be speaking later, but also diabetic retinopathy, VHL, McTel, and many other diseases. I'm very pleased and honored to present this year's Proctor Medal to Emily Chiu. Emily, please. Next, I would like to introduce Dr. Lloyd Paul Aiello, Professor of Ophthalmology at Harvard Medical School, and he will review more of Emily's achievements and, of course, introduce her lecture. Lloyd, yeah.
Thank you, Tian. It is an exceptional thrill for me today to introduce the 2024 Proctor Award lecturer, Dr. Emily Chiu, an extraordinary individual who has been collaborator and mentor to so many of us around the world. The Proctor Award honors outstanding research in basic or clinical sciences as applied to ophthalmology, as you just heard. And Dr. Chiu is clearly one of the most accomplished individuals of our lifetime in this regard. During her long and diverse career, she has designed and conducted and reported some of the most important large clinical trials, as you just heard, on a variety of topics. She has published over 540 papers, the impact of which are seen by an H-index of 121, meaning that nearly 1/4 of her patients have well over 100 citations each. Her total citations as of yesterday exceeded 80,300. I did not check today because I thought I'd run out of room on the slide. She has published 21 editorials, 32 book chapters. She's been on no less than 27 NIH committees and a member of 8 editorial boards. But when Emily undertakes any of her projects, she always does so at the highest level. So she's not just been on the editorial boards, but editor-in-chief of 2 major journals and president of some of our most prestigious organizations, including the one here today, ARVO. Over the years, she has obtained 46 awards. Only a small list are shown here, but clearly they include our profession's highest honors. She has also given 28 named lectures. Both of those numbers will go up by 1 at the end of this session. Her smile and enthusiasm have been present even from the early studies, initially in the University of British Columbia, then the University of Toronto, all the way through residency, and then the fellowship at Johns Hopkins, where she worked with none other than Irene Monami and Arnold Patz, and then in the Netherlands with August Doetman. She then began her storied career at the NIH, first as a visiting scientist in 1987, around the time I first interacted with her, and then working her way up to her present roles, as you can see here, and as the director of the Division of Epidemiology and Clinical Applications, as well as the deputy clinical director at the National Eye Institute. Now, the most obvious but there's far more to Emily than just the accolades and so forth that you've seen here. The most obvious, of course, is an incredible smile that she has, and it's no doubt that with this, the National Eye Institute considers herself one of the vision leaders, not only in the US, but worldwide. I used to think that there was no picture of Emily that didn't have her smiling, and that was true until just recently. We had to go back, way, way back to Canton, China, when she was born, shown here with her mother. And I'm sure she would have been smiling if she wasn't told that this was a strictly formal portrait. But ever since the smile's been present, here with some of her mentee families, she has mentored more than 86 individuals for a year or more, some of whom are in the most prestigious academic positions and other positions in vision science around the world. And then, of course, there's her own family, all of whom, as you can see here, share her joy of life. The three girls, Allison, Emma, and Erica, and her wonderful husband, Dr. Bob Murphy, who himself made major contributions to academic ophthalmology, especially during the early years of diabetic retinopathy and laser photocoagulation. Now, just in case you were wondering who brought the smiles to the family, I do have a slightly earlier picture, and here you can clearly see that the girls and Bob are practicing their smiles, and Emily is in the middle doing the mentoring. Of course, everyone is growing, with both Allison and Emma now being medical doctors and Erica on tenure track at the University of Syracuse in the drama department. And there are two grandchildren, Rosie, 4, and Maddie, 2, both just starting their own journeys, but doing so only a half-mile away from Bob and Emily. Emily has many other talents as well, and just one is being an exceptional cook. Whether it's making the wedding cake for her family or setting up what she refers to as nothing much, just a bit to eat. You know that any meal that she prepares will be incredible, and you will be met with the incredible kindness, the infectious enthusiasm, and that smile that is Emily. I can also completely assure you that the food will be some of the best you've ever had in a private residence, and that is absolutely true, but I can only tell you that tonight because my mother is not in the audience. I cannot be more sincere when I say that it is a true honor to be able to call Dr. Chu a mentor, a colleague, a collaborator, and a friend, as so many of us do around the world. And now it is one of the greatest privileges of my career to be able to introduce the 2024 Proctor Award lecturer, Dr. Emily Chu.
Wow, that was quite an introduction. Thank you so much, Lloyd Paul. I really appreciate your wonderful sense of humor. We all need it. And thank you very much to ARVO for this wonderful privilege and honor to be a Proctor lecturer. ARVO's been very, very important to me. As a medical student, I was brought here as my first-year resident by my mentor, Brenda Galley. And it changed my life. I've been here every year, except for the year when I had my third child, and one child was sick. So that was 2 years that I've missed, but I've been here ever since. It's a great society. I've been very honored to serve and to represent it all over the world. And what's also really interesting is the fact that we had great leadership. Iris and Justine looked at the membership demographics, and we started a women's leadership program, which I'm very proud of. And last decade, or last century, we had 0 women leadership. But now, this current century, the last 24 years, we have 28%, which is fantastic. We still have a lot more to do. Diversity is really important in science. It makes our science better. It improves our productivity. And we have much more to do with URMs. And so let's every one of us do our part to make sure that's true. Thank you, ARVO, for shining light on NEI. And I'm really pleased that I'm sharing this with my colleague, Anand Swarup, which is really, really joyful. And so now to talk about the lecture itself, I have no financial disclosures. I'm talking about AMD and the epidemic we are tackling and how we can actually do this in the next couple of decades, we hope. Just to give you an idea what I'm talking about, I want to be on the same page as early, intermediate, and late AMD. In the past, we called neovascular and just central GA and the arid circulation and our classification. We now have GA anywhere as also being one of the outcomes. Macular degeneration is indeed a leading cause of blindness. It's mostly in the high-income countries. It ranks really third in the world for blindness and accounts for 9% of all blindness. And it's predicted by 2040 to affect 288 million people worldwide. So that's a huge, huge number of people. We know from a lot of population-based studies based in America, in India, in China, and also in Europe and Australia, that the advanced AMD increases with increasing age. Interestingly, two really icons of our field and who were very involved in ARVO were Barbara and Ron Klein, who suggested, in fact, the incidence is actually going down. The greatest generation are those who are the oldest. As you go down to the baby boomers, the incidence is actually going down. That may be because we're doing better with our lifestyle. We're smoking less. We're having improved lifestyle. But the incidence of AMD is decreasing. On the other hand, the World Health Organization estimates that by 2050, that we're going to have a doubling of people over age of 50, from 1 billion to 2 billion people in the world. So the risk of getting macular degeneration is going to increase, and in fact, it's going to triple for patients who are 80 years old, 427 million people. So that's a large number of population that we have to deal with. So what can we do? Prevention, prevention, prevention is really, really important. Let's talk some more about prevention. And we've done this at the National Eye Institute for a number of years. My good friend and colleague, Rick Ferris, who chaired the first ARIS study, did the study. We did a clinical trial in 1992 to 2001. And then 2001, 2005, we had a follow-up study. Studies from epidemiological studies suggest that nutrition is very important for macular degeneration. And we looked at vitamin C, vitamin E, beta-carotene. We added zinc and copper as it was tested just prior to this. And much to our surprise, we actually found that this supplement can reduce the risk of progression to late disease by as much as 25% in 5 years. And it's really interesting we did the data, but it's nice how other people actually look at it and replicate our data. So the Cochrane systematic review was done just last year, looking at a number of countries, 4 different countries, 26 studies, and 13 of them actually qualify, and this is what they found. You can see ARIS is on the very top. It's got a big block, red block. I'm just going to show you a little bit larger. That means the point estimate means that there's protective effect. It's to the left of 1, and the whole conglomerate, the whole aggregate, and that diamond shape shows that there is indeed evidence suggesting that ARIS supplement does reduce the risk of progression to late AMD. And if we look at the data from year 5 and we follow them, you can see that it's just a very jumbled up slide, but if you look right in this area, it's particularly important for neovascular AMD. Supplements are very important for neovascular AMD for reducing it. It's good for late AMD in general, but neovascular in particular. And when we look at 10-year data, these are patients who finished the study at 5 years. We started giving them the supplement, so they're all taking the supplement. Even at 10 years, we can still see a difference, that the neovascular AMD is still protected by at least 25%. So that's pretty encouraging. But we then went on to do another study called ARIS-2. ARIS-2 is a study that was brought on in 2006, and this is a clinical trial, another clinical trial, large randomized trial. The first one was about 5,000 patients, and this one's about 4,203 patients, and we looked at those patients for 5 years, and then followed them for an epi study for another 5 years. So this is a long-term 10-year study. ARIS-1 was also a 10-year study with lots of data to really mull over. And why did we do ARIS-2? Well, because of data from ARIS-1. So this is the food frequency questionnaire. We asked our patients for the past year, what did they eat? We did that in ARIS-1. And ARIS-2, we did the same thing using different, what we call food frequency questionnaires, asking the patients what they did. And this is interesting for me because as a young child growing up in a Chinese immigrant family in Vancouver Island, our mother would actually give us the goji berries and said, it's good for your eyes. Well, it's filled with lutein. And our dad, before a very hard exam, he'll say, you need brain food. We're going to give you a fish tonight. And in fact, those are the 2 things I ended up studying. So how would they know that I was going to study it in my adult life? So in ARIDS, when you look at the baseline of this, you see that neovascular AMD, geographic atrophy, even large drusen are protected in association. This is an association, not an incident study, but again, it's protected. But if you look at omega-3 fatty acids, if you eat fish just twice a week, you have a reduction in neovascular AMD. Ironic acid is from patients eating a lot of red meat. That's a line to the right. So fish is really, really important. If you look at instant disease, looking at patients who are progressing to neovascular AMD or geographic atrophy, that's also protective. So we have data suggesting both at baseline association and looking at incident new data that new cases were also protected. So that is the data that suggests that we should look at lutein and zeaxanthine for ARIDS2. So also, we have basic biological plausibility. Lutein is found in the macula. That yellow streak you see there is very important for the actual macular pigment, and the fact that omega-3 fatty acids are a really important part of the membrane, which makes up a lot of the retina. So for those reasons, we embarked on a new study, ARIDS2. In this study, if you're an Asian, you'd have goji berries, but you would have spinach or collard green or kale. That's what we found that to be really important. So this study looked at adding lutein zeaxanthine, 10 milligrams, 2 milligrams of lutein and zeaxanthine. That's what naturally occurs in life in terms of the fruits and vegetables that we see. Omega-3 fatty acids were DHA, EPA, 1 gram, and this was adding on top of the ARIDS formulation. The patients we studied in ARIDS2 were more severe. They already have intermediate AMD, and we want them to be on ARIDS supplement. So now we're looking at adding these two factors to see whether we have any effect in reducing the risk of macular degeneration. And in fact, both studies looked at cataract as well. We don't speak much about the cataract. We really didn't find much of effect, but it was on aging disease as we're talking about. So what did we find in the first study, in the first 5 years of the ARIDS? You see that the primary randomization looking at fatty acids, the DHA, EPA, there was no beneficial or harmful effect. But lutein zeaxanthine actually increased, benefit was an incremental increase compared to the placebo or the control group. And we also found that if patients who don't eat any greens at all, those are the bottom line of the people who said, I never touch spinach, I don't eat ever any green, and there are people like that. I have patients in my study and in my practice who tell me I would never eat those vegetables. And there are 25% treatment effect in those patients. So clearly that has an impact. And so when you do direct comparison between lutein zeaxanthine, remember we had beta-carotene in our ARIDS1 formulation, but in ARIDS2 we actually could look at them head to head. And lutein always went up between that and beta-carotene. It was always better than beta-carotene. But what was even more striking was that beta-carotene increases the risk of lung cancer. Let me just go back, and I go back. Okay, no, I don't have that slide here. Sorry, there's one slide missing, but it doubles the risk of lung cancer in patients in beta-carotene. So for that reason, we were very concerned whether long-term effects might persist. So patients were then followed for 10 years. Patients were given ARIDS2 supplement, no longer any control group. Everybody's taking the ARIDS supplement, and we followed them for another 5 years. So 10-year follow-up. And the long-term effects, again, you'd see on the very top are the lutein zeaxanthine. The actual point estimate is about 10% improvement over no lutein zeaxanthine. But if you look just directly, and this is now very similar to what we found in ARIDS in the first five years of the ARIDS2 study. If you look comparing lutein zeaxanthine to no lutein zeaxanthine, which is on your left, on your right, looking at those without beta-carotene, you can see that there's a difference. There truly is a difference between the two of them. And at 10 years, we replicated what was seen in the ARIDS2 5-year follow-up. We found that it almost doubled the risk of lung cancer. And these are former smokers. So if you're a former smoker versus non-smoker, you double the risk of getting lung cancer. So for those reasons, we had switched beta-carotene and used lutein zeaxanthine. So we now confirm that lutein zeaxanthine is indeed a very safe and better choice for the ARIDS supplement. So for that reason, we have that ARIDS2 supplement. Now, this is and I want to diverse a little bit now and talk about the actual diets. And diets is really important. And food really is medicine. And you can see here, this is from one of the papers looking at particularly a randomized trial of Mediterranean diet for cardiovascular disease. This was done in 7,000 patients in Spain, and they found that there was as much as a 30% reduction in cardiovascular disease. So this is a first really clinical trial of the diet, which is really important, showing that you are what you eat. And right now, the government, our government, I work for the government, actually really is very interested in nutrition. And the White House actually has had a conference on hunger, nutrition, and health. We finally realized a lot of our health issues are due to disparities and problems with nutrition. And in fact, food as medicine is one of the subjects that we're actually approaching with the White House as well. So the research question we have is, what is the association of Mediterranean diet with AMD? There have been previous studies that have been looked at this, on the Mediterranean diet. We used the ARVO data, which showed you that Dr. Seddon and others looked at it, that there was a beneficial effect. And there are other studies by our colleagues who have worked in all over Europe, both the association studies and longitudinal studies, show that there is a beneficial effect of Mediterranean diet by at least 25 to 40%. So we embarked on looking at this in our data as well, too. So I have these great colleagues at NEI, as well as our extramural folks. Dr. Julie Mares from the University of Wisconsin was one of our consults. Dr. Tunan Keenan and Elvira Agron, who's our wonderful statistician, have delved into this. And we looked at and these are two papers that we talked about. And these are really, I think, interesting data showing that indeed diet can be very important. So just a word about Mediterranean diet. There's 9 components to that. And mostly it's high in fruits and vegetables, legumes and nuts, and just very moderate in wine, moderate fish and white meat, and really monounsaturated fatty acid is really important. This is particularly important in the Europeans, where they use a lot more olive oil than we do in America, and also very low on red meat and refined sugar. So when we looked at that diet, and we could, from our dietary questionnaire, we found that when you look at patients looking at progression to late AMD, either neovascular or geographic atrophy, everything's to the right and left. That means it's protective. You can see that for late disease, for geographic atrophy, neovascular AMD, it is protective. And what sort of numbers are we talking about? Well, these are the hazard ratios. That's 0.71 for geographic atrophy, and 0.89 for neovascular AMD, and 0.77 for late AMD. This translates to really a 29% reduction for geographic atrophy, and really a little less for neovascular AMD and less for combined. But clearly, the advanced AMD is protected. So if you already have intermediate AMD and you have a good diet, you can actually stand a chance of reducing your risk of late AMD. And so this is, I think, important for us as clinicians to make sure that we get that to our patients, that they understand that. And if we look at the different components of the actual dietary questionnaire, I talked about the 9 components. What really drove it was fish. So fish twice a week is what we say a high intake. The hazard ratio is 0.69, p-value of 0.001. This is a pretty stringent test. We do a Bonferroni, which for you who don't understand this, is that we take the p-value and divide it by a number of tests we do. It's like you put your hands too many times in the cookie jar, you have to get a penalty, and the penalty is dividing 0.05 by whatever numbers you have. And so it could be very stringent, and we found we actually achieved this, and so it's quite evident that fish is really important in terms of reducing geographic atrophy and late AMD. So fish is a component of the Mediterranean diet associated with 31% decreased risk of progression to late AMD, to geographic atrophy. And for those patients who don't have AMD, they have either early AMD or no AMD, what is their risk of developing large drusen, which is a sign of intermediate AMD? And here you see that the forest plot does not touch 1. The estimate, which is the black spot, is about 0.75, and you see that this hazard rate of 0.75. So even if you don't have AMD, you have a reduced risk of having late AMD, or large drusen rather. So higher adherence to the Mediterranean diet is associated with decreased risk of progression to large drusen. And what's really interesting is that we can reduce your rate of getting geographic atrophy with the diet, but if you have geographic atrophy, this can also help you as well too. Patients with high adherence to the Mediterranean diet had a lower rate of progression of the geographic atrophy. You see on the left that this is the fundus autofluorescence showing that the spot grows over time, and that is being measured. If we can slow that growth, that could be very important. And lo and behold, when we looked at the actual diet, the highest intake of the Mediterranean diet it has a slower growth rate than the other two. So that clearly is an important outcome for us to think about. Even if you get geographic atrophy, you can slow the disease down by this. This is the brainchild of Dr. Jernan Keenan, which is a really interesting data, which we're now trying to get this published. This is hot off the press data we're looking at. So geographic atrophy seems to, in 2/3 of our cases, they don't start in the center. 1/3 will start right in the center and cause problems right away. But 2/3, you actually go around the center. You preserve that macula, the fovea, is preserved to the very end. And when you look at the growth of the lesion from the center of that macula to the closest area where that geographic atrophy is encroaching, you can see that over the few years of this patient, that it decreases. So that number is called proximity-based evaluation that we're looking at. So when you look at that, for AREDS1, we looked at antioxidants versus none, and you can see that there is actually a treatment effect of about 36% in reducing the progression of geographic atrophy towards the center. Well, that's pretty significant. In AREDS2, we found similarly. Now remember AREDS2, everybody's taking the beta carotene already, so they're already on it. On top of that, we gave them lutein zeaxanthine. We look at the lutein zeaxanthine. In the first row, you see that there is also a treatment effect. The progression rate is much higher in those not taking lutein zeaxanthine versus those taking it. And the second part, the lower part of that graph, shows you what happens when you look at beta carotene that's not been assigned to them. So in other words, we're really looking at really lutein zeaxanthine without the beta carotene. You can see that there's a treatment effect indeed. So this is brand new, and this is something we need to investigate further. What do we do with this information? We need to do further investigation to understand that better. So just to recap what I told you so that you won't forget this, because this is important. AREDS1 and AREDS2 supplements reduces the risk of progression to late AMD, especially neovascular AMD, by 25% in 5 years. And post hoc analysis demonstrate that AREDS and AREDS2 supplements reduce the rate of proximity-based progression in non-central geographic atrophy. Again, that needs further information to be investigated. The Mediterranean diet is associated with decreased progression to large drusen. That's people who don't really have intermediate AMD. They're actually predicted about 25% reduction to that. And decreased progression to late AMD, especially for geographic atrophy in those who had intermediate AMD. The effects appear to be both early and late AMD in those, so it really is important to look at this. In addition, we've showed that the diet itself can be showed to decrease progression to geographic atrophy, but also a slower rate of the progression of the GA lesion itself. So it's probably never too late to start a diet, and associations, of course, but not results of a clinical trial. But we do have a clinical trial now, and this is done by Dr. Carolyn Klaver. She is doing one in the Netherlands. She has started this, and it's a standard, really looking at 3 prongs of this and seeing whether they can actually have this diet, which diet randomizations are difficult, as we all know. We've done some in diabetes, and they don't always stay, but this is really important to look at. And she's looking at standard therapy, and then looking at standard therapy, plus looking at the risk profile, looking at whether you're at genetic risk or not, and then looking at adding a coach to this. So this is going to be an interesting trial to watch. And we also have an application in from the REMER, the Ryan Initiative Macular Research Group, looking at basically an operational way of doing this, and we've actually been granted this, and this will be started soon as well. Well, and then I go to see my friend Anand Swarup and said, how can we explain these issues? And he has his wonderful postdoc, Dr. Anupam Mondal, and we have great colleagues at NHLBI who are the world's experts on lipids in cardiovascular disease. So NHLBI, Dr. Yang and Dr. Ramali. So between them, we actually did some more studies looking at what happens over time. And this is, these are animal studies, obviously. These are aged mice who are 12 months old. They're given either a control or an ARID supplement, well, in the supplement, in their diet, of course. They're not going to give them capsules. And we'll see what happens over 6 months. We also have another group of mice who are then randomized to Mediterranean diet and a really high-fat diet. And these really interesting results show that the principal component analysis show that there are 6 differences in the genetic response to this. So this is interesting. I understand that other people see that as well. So this is very different in terms of the female and the male respond differently, and different genes are activated. So that needs further investigations to see what happens with that. And what's really interesting with the ARID supplement is that it also activates different sets of genes. And one of the important mechanisms is actually with fatty acid metabolism, which is quite high. In the lower bar, you can see that there are a number of abundant fatty acids that are elevated in the mice who are given the ARID supplement diet. And you can see in the final bar with the bright red and the blue that there is an increase in omega-3 fatty acids in the retina of those given ARIDs. So clearly we're looking at some way of explaining some of the reasons why we see the effect that we see in ARIDs. Now the Mediterranean diet is also interesting in that it increases, the high-fat diet really is something that we probably, a lot of us eat in America, but the Mediterranean diet really is important. But the top pathways that's enriched in the Mediterranean diet include the pentose phosphate pathway, the PPAR signaling, and the fatty acids. So fatty acids is a recurrent theme we hear over and over again, even though we didn't see an effect in ARIDS2 when we gave the DHA and EPA. And also the transcriptome on the very bottom there shows that the mTOR and metabolic signaling respond differently to these isocaloric diets that were given to the mice. So what do we do next? Well, what's the direction? Integrating the omics signatures that identify retinal mechanism of ARIDS2 would be very important. Identifying factors that influence retinal adaptation is important. Examining the retinal function with ARIDS2 treatment and other complex diets is also important. Benchmarking retinal adaptation with other key organs will be in order to be done. So you are what you eat, and food is medicine. And currently, the ARIDS1, ARIDS2 has a project called the Omics. Again, Tiernan and Claire Weber and our great statisticians, and we have wonderful help from NCATS. Dr. Evie Matthew and Dr. Krista Clare are really members of our team. We've gotten support from the Office of Dietary Supplement, Office of Women's Research, Health Research, the NCCIH, as well as NCATS and IEHs are all in collaboration with this project. So what exactly is this? So, ARIDS1 and ARIDS2 is like this gift that keeps on giving. Although we stopped the study a number of years ago, 5 years ago for ARIDS2, we collected blood on an annual basis. So you can see that over time, we started with 1,588 patients, but by the time of year 6, we have almost 1,000 patients with blood. This is ARIDS1, and these are fasting bloods. And ARIDS2, we have about 500 patients who have gone through this, and they've done every 2 years. So we have all these serum that's been collected, and we're very interested in looking at the metabolomics. This is Dr. Anneke van Hollander's great graph showing what some of the things we like to do. So most of this will be looking at metabolomics. The NCATS collaborators are very interested in proteomics. They're actually measuring proteomics for us. We're also doing the genomics. We have whole genome sequencing on this. And we also have some funding from Women's Health to look at the actual DNA methylation. So clearly this is a project that involves many of us, and we hope that this will give us some answers that we're really interested in seeing. These are the results of the metabolomics. We're looking only at the baseline. I'm storing this very small numbers, but you see top ones for ARIDS1 and ARIDS2. And here are the pathways enrichment that are common to both. And we hope that we will get more data and be able to write this up in a very short order. And also looking at the chemical class. These are the common chemical class between ARIDS1 and ARIDS2. So we're very, very hopeful that this project will give us some more answers. And of course, I have to pay tribute to all my colleagues who have done incredible work in this area already. What's interesting about these 3 studies is that it really points to fatty acids as being very important. So again, that's a recurring theme. This is very important. And this is a study done by Dr. Claver, and again, 94% of them are in the intermediate group is related to lipids. So lipids play a very important role. So what we hope to do is looking at progressors, non-progressors, data on genomic interaction, data on early intermediate AMD will help focus on the early prevention. And gaps in knowledge are natural history of early AMD that we hope to get more on. New targets may be identified through new metabolomics, proteomic, genomic studies. And dietary animal studies will also be very important. And just to really emphasize the importance of long-term follow-up, ARVO-1 and ARVO-2 were 10-year follow-up. In addition to that treatment effect, we learned a lot about the natural history. So for example, these are patients who were evaluated by my colleagues at Belfast and University of Wisconsin, Dr. Dombatelli and Tunde Pito, showing, look at the striking differences between 5-year and 10-year. You see that there's tremendous progression in the geographic atrophy. It's not only in the macular area, it's in the peripheral area. So it's like a pan-retinal disease we're talking about. And we see this over and over again. So these projects are really important for this long-term follow-up. Another important aspect from these 10-year follow-up is that we identify an important risk factor called reticular pseudodrusen, or RPD. And here, the reticular pseudodrusen is different from the typical drusen you see in macular degeneration. Instead of being underneath the retina, it's actually above, underneath the RPE, it's above the RPE. And Christine Curcio has done some great work in this and showing in her histopathology slides there. And this is really an important risk factor. And we call it the 3rd macular risk factor because it's important in identifying this. We want to help our doctors to identify patients who are at risk. We have a daunting number of people who are going to get macular degeneration. How do we help them? And this is a photo showing that the reticular pseudodrusen's above, and down below you see large drusen. And Dr. Kathy Kukris, one of my colleagues, have shown that it's really important to identify them because it really signal a functional deficit as well. There's very poor dark adaptation. We also identify this RPD as being important for geographic atrophy. If you have this risk factor, your geographic atrophy, your dry type macular degeneration, will actually progress even faster. And in addition to that, we know that it's also important for late AMD as well. And the best way to diagnose it, of course, is with OCT. Here's a patient of mine from AREDS who's 97-year-old. I've followed since the beginning of 1992. He has RPD, but he looks perfectly normal. You can't see it there. But when you look at the near infrared, you see this. And of course, the OCT, you can actually see this. So this is one of the signs that's very difficult for doctors to actually diagnose. And just going into the studies that we've done in the past, looking at risk factors for macular degeneration, we found in AREDS that if you look at just drusen and pigment, you can count on your hands whether the patient's at risk or not. And if you have all 2 in both hands, you are at the maximum at 4. And there's a 50% chance in 5 years you'll progress to late AMD. If you have none, your chance is very low. So this is an easy, simple scale that we clinicians use all the time. But now we have RPD, reticular pseudodrusen, which changes that formula a little bit. You can see that with RPD, instead of 50%, it goes up to 72%. Instead of 27%, it almost doubles. So it's important for us to understand how to diagnose this, and this is one of the things we want to talk about. The other part of the equation that we want to talk about, looking after these patients, is the fact that there's a shortage of physicians. How many of you try to get just a general practitioner? I mean, I have troubles finding one, and I'm a physician. And it's going to get worse. In 2036, so these are very complex analyses that are being done. This is done by the American Association of Medical College. They look to see what's going to happen over the period of time. So by 2036, they may be as high as 86,000 doctors that are deficit, or as low as 26 or so. So that is a large number of patients. So what can we do? How can we help this whole situation in terms of making macular degeneration more accessible and more easily diagnosed and then treated, and patients being monitored? So of course, medical artificial intelligence is our next step, which is really important. As you know, it's part of our lives already. And ophthalmology is actually leading the way in all the work in artificial intelligence. So we should be doing this to help our patients in the future. We have a great team from NLM, our National Library of Medicine. These are my collaborators, as well as the friends from NEI themselves. And just to give you an idea, I want to make sure that you're all aware of the fact that we share our data as part of what we're doing in research. And you can see here that the sets of data from images from 10 years, which are taken annually, this is nearly 60,000 images. They're not just any images. These are curated images. These are images that have been read by the graders and adjudicated, looking for all sorts of things. So these are very rare to have such a well curated. And the fact we have the metadata with it, we have all the medical data. We have information on their cardiovascular disease. What did they see that day? We also have cognitive function testing, dietary questionnaire. So this is a very rich data set that we have. And certainly we've been doing works on this, and one of the deep CNET, which we use is an algorithm that we looked at for actually classifying AMD. And these, of course, are available on dbGaP. In AREDS 2, we have 4,000 patients who've been seen for 5 years. Now we've actually have some diversity in images. And multimodal images will be very important for our future analysis. In this case, we have fundus autofluorescence in a good portion of those patients, and these are also very important for future research. And thanks to Cynthia Toth, who was a real pioneer in getting a sectoral domain OCT started. And this was a long time ago, and we only had bioptogen, but we have a number of OCTs as well. So these really are very important for our work in terms of artificial intelligence. We also have lens photographs on every patient for 10 years annually. And these have been used by a number of people. And these have been downloaded by a number of researchers as well. So I just want to talk about reticular pseudodrusen again because of its importance and how can we actually detect it? How can we help the doctors in the future to find this important risk factor? And this is a study done with our collaboration with our computer scientists, looking at how we can evaluate this. And in our case, this is color photographs and fundus autofluorescence. And this is using what we call a 3M, it's multimodal, multi-attention. And this is looking, again, at these patients on how to really develop a framework for deep learning to look at reticular pseudodrusen. This multitask means that we're using different tasks. We're actually looking at geographic atrophy and hyperpigmentation at the same time, so we look for reticular pseudodrusen. We're looking for different features for representation of the actual operation itself. And then with the performance of the three RPD detection was compared with the state-of-the-art deep learning models that are out there. And this really was very important for looking at how we as ophthalmologists look at this. Can we compare it to the ophthalmologist? We also looked at the detection of geographic atrophy. So there are other procedures we put into there to see how well it would actually function. And furthermore, we validated it with the Rotterdam Eye study. And this is the workflow of the actual work that's been done, going through. I'm not going to go through all the details with you, but the final analysis shows that the color photographs and the OCT together had a better accuracy and were able to actually detect on even color photographs in patients who don't have OCTs. So this is important, especially in areas where we don't have an OCT available to everyone. So the M3 performance was superior to that of the deep learning methods of any human experts. Performance was twice as high as that of a retina specialist. So we're trying to help the retina specialists. And really, this M3 approach is highly generalizable. External validation was excellent using the Rotterdam Eye studies that I mentioned. And RPD on color photographs alone was actually very successful. So generalizability also shown by adaptation to deciding two other functions, such as looking for GA and hyperpigmentation. And we have a number of other studies. A number of people have already done a lot of work in this, and many of you in the audience are experts in this area, but it's just really an important area that we're going to look into. But that's not going to be so easy. Overall, these are, the M3 is very important, but we need to test it and see whether we go into clinical trial. So one of the studies that we really were interested in looking at, and this is from the collaborative community on ophthalmic imaging, which is supported by the FDA, as well as a number of stakeholders. We were really looking at how do we go from data to actually implementing these algorithms into actual practice, both for clinical trials, for implementing for healthcare itself. And this was published with our group of colleagues. And what's the pathway to use AI? How do we incorporate it into our clinical practice? And for clinical research, such as clinical trials, it might result in an FDA-authorized and AI-based device for clinical care. There are multiple steps that are needed, and there are no FDA-authorized AI-based device for AMD currently. There are tons for X-ray and radiology, but not so much for us, so we have a bit of catching up to do. And we really need education of the users and the patients and also the institutions themselves on how to do this. It would benefit providers, the healthcare system, and the patients. The impact on clinical trials would be actually tremendous when we get to this stage. So these are some of the steps that we need to think about, getting who do we use this for? The clinical use, what would that be? Collecting the data sets have to be curated and really understood and really made available, but also very equitable in the fact that we have to do it in a population that will reflect the people that we're going to use it on. So it's all done in a white population, but we go to Asia with it. Will that work? So we really need to get all these things in order and eventually go through all the validation that is required for this device. So acquisition and curation of large, high-quality data sets is difficult. Dr. Damapalli is actually going to do an excellent one where it's sponsored by University of Wisconsin, looking at a validation set up for AMD. A development of architecture, AI architecture, and the training and validation of model include functional interaction with the model and output in the clinical end users. So we have to make sure it actually works in that case. The impact of this, we have to evaluate the impact and whether it really helps the physicians, what does it actually do. So there's much more to be done in this field. And we could use it either for screening, and there could be color photographs or OCT for diagnosing, and also for managing, and obviously my desire, of course, for clinical trials. We can work with clinical trial. So these are really important steps in terms of working towards the FDA-approved algorithm that can be used for actual research as well as for clinical practice. So in summary, I've presented to you secondary prevention with AREDS2 supplement for intermediate AMD and for an extension beyond, and that needs to be evaluated further. The importance of diet is something we have to get that message to our patients. Food is medicine, and the fact that the government's really interested in now, and we may look at the food deserts and how to get those people who really need the food, and how do we make it more equitable. Potential targets from future metabolomic and proteomic and genomic studies will be very important for us to think about what our next step might be. We also need natural history of the very early AMD cases as functional changes. How do they actually occur? We're still missing that piece in our AMD armamentarium. Potentials for an AREDS3, can we do it at prevention and prevention of early AMD? Prevention is much better than trying to go to the late stage. Future research and implementation of medical and artificial intelligence in diagnosing, monitoring, and predicting progression of AMD, and for helping us with clinical trials would be tremendous. There are other gaps in knowledge in care of macular degeneration. We need better and more durable therapies for neovascular AMD. Working on that. We're very fortunate we have 2 wonderful drugs that have been FDA approved, but we need more efficacious drugs for GA, and that needs to be worked on as well, too. And we need other innovative technology being tested, such as stem cell work, gene therapy for the late stages. The prevention of the early stage would be important, would reduce the need for the more burdensome and expensive therapy needed in the later stages. They're often, and may have very dire complications from. They're rare, but they can happen. So there's a large burden of development care. The medical artificial intelligence will be really important as we try to alleviate this burden of care. So prevention is indeed very, very important. I'd like to thank the ARVO investigators who were really a big part of this, especially John Paul Sangiovanni, who passed away, one of our major movers and shakers in our study, the ARVO-2 investigators, and most of all, the ARVO-1 and ARVO-2 patients. They spent hours with us and over a period of 10 years. That's real devotion. We're very appreciative of that. And my collaborators, who past and present at NIH and beyond, they've been a real rock to helping us with all the work that we've done. I'd like to give a shout out to all my NEI colleagues because they really inspire me because every day they are there giving service, sacrificing, and doing immense work. So kudos to NEI itself, and we're so happy that ARVO has actually shone this light on NEI. We're very grateful for that. I can't thank all my mentors enough, and there's so many of them. I'm just going to talk about institutions. Some of them, there are many other institutions who were so, these are the Hallow Halls that I actually visited. They were very important in my career and my life. I'm very grateful to them. And most of all, I'm grateful to my family. They keep me grounded, and they tell me what's really important in life. And thank you all for coming, and that's in my email. If you have any questions, please ask me.